Determining the matrix polysaccharide composition and crystalline cellulose content of plant cell walls starts with dried plant material, which is subjected to various extractions. The resulting material consists of cell walls only and then is split up. To determine the matrix polysaccharide composition, the wall material is treated with a weak acid to hydrolyze non-crystalline polysaccharides, which are mainly the hemocelluloses, into their monosaccharides. The monosaccharides are then derivatized into their corresponding volatile alditol acetates, which are then analyzed and quantified by gas chromatography coupled to a mass spectrometer. For the determination of crystalline cellulose content, walls are treated with the Updegraff reagent, a mixture of acids that strip away non-crystalline wall components. The remaining crystalline cellulose is degraded into its monomer glucose, which can be assayed colorimetrically. The determination of lignin content and composition is demonstrated in another Jove video publication entitled Comprehensive Compositional Analysis of Plant Cell Walls, Part 1, Lignin.
Hi, I'm Marcus Pauli, head of the Wall Analytical Facility at the Great Lakes Bioenergy Research Center here at Michigan State University.
I'm Cliff Foster, manager of the Cell Wall Analytical Facility. Today we'll show you how to determine the composition of the polysaccharides present in the plant cell wall material, that is cellulose and various hemicelluloses.
Here at the Great Lakes Bioenergy Research Center, we develop technology to convert these sugars into biofuels. In order to identify an optimal plant feedstock, we first need to assess the kind and abundance of sugars that is present in this biomass.
In another video entitled Comprehensive Compositional Analysis of Plant Cell Walls, Part 1, Lignin, we will focus on the polyphenol analysis. So let's get started.
Begin this procedure by isolating cell walls. Grind roughly 60 to 70 milligrams of dried plant material with 5.5 millimeter stainless steel balls in a 2 ml Sardsted screw cap tube using a Retsch mill. As a high-throughput alternative, use a grinding and dispensing robot termed iWall as described in Part 1. When grinding is complete, remove the steel balls. Continue to isolate cell walls and remove residual starch as shown in Part 1 and described in the accompanying written protocol. Weigh out 2 milligrams of de-starched cell wall material into a 2 ml Sardsted tube. A high-throughput way of performing this task is the iWall robot. The robot takes a tube containing ground plant material and pokes a hole in the bottom of the tube. The pierced tube is then placed on a vibro feeder. An empty tube is placed on a balance and shifted under the vibro feeder. Through vibration, material percolates through the hole in the loaded top tube into the empty bottom tube until the desired amount of material is accumulated. Add 100 micrograms of inositol as an internal standard. Rinse the tube walls with acetone to collect the cell wall material on the bottom of the tube and evaporate the acetone very gently under airflow. Next, perform weak acid hydrolysis. Carefully add 250 microliters of 2 molar trifluoroacetic acid, or TFA, to each sample, making sure no material is splashed up onto the tube walls. Cap the tubes tightly and incubate at 121 degrees Celsius in a heating block for 90 minutes. Then, cool the heating blocks and samples on ice and centrifuge the tubes at 10,000 RPM for 10 minutes. Transfer 100 microliters of the acidic supernatant containing the solubilized sugars of the matrix polysaccharide to screw cap vials, making sure not to disturb the pellet material. The pellet can be used for the crystalline cellulose assay. Evaporate the TFA in a glass tube under a gentle stream of air in an evaporation device. Add 300 microliters of 2-propanol to remove excess acid, vortex, and evaporate at 25 degrees Celsius. Repeat for a total of 3 times. In order to enable identification of the monosaccharides by gas chromatography, perform an alditol acetate derivatization. First, reduce the monosaccharides to their corresponding open ring alditols, which are then acetylated. Consult the accompanying written protocol for detailed instructions. Each step of the procedure follows the previously shown protocol: addition of reagent, incubation, evaporation under a stream of air, followed by removal of excess reagent by various washing steps. In the final steps, the alditol acetates are extracted. First, add 500 microliters of ethyl acetate and swirl lightly. Then, add 2 ml of water, cap the tubes, and vortex. Centrifuge the tubes at 2000 RPM for 5 minutes to obtain clear separate layers with ethyl acetate on top, containing the alditol acetates, and water on bottom. Pipette 50 microliters of ethyl acetate layer into GCMS vials with inserts. Dilute by adding 100 microliters of acetone to the GC vial and cap. The GC vial can be stored at 4 degrees Celsius if the GCMS analysis does not immediately proceed. Inject the samples into a gas chromatographer that is equipped with a quadrupole mass spectrometer. For column and details of the run, please consult the accompanying written protocol. The starting material for determining the crystalline cellulose content can be isolated cell wall material or wall material already treated with the weak acid TFA, either immediately after acid treatment or in a storable dried form after removing excess TFA with isopropanol, as shown previously. Add to the TFA pellet in the 2 ml Sardsted tube 1 ml of Oop de Graaf reagent made up of acetic acid, nitric acid, and water at an 8:1:2 volume-to-volume ratio. Cap the tube tightly, vortex, and heat on a heating block at 100 degrees Celsius for 30 minutes. As a result of this treatment, only crystalline cellulose remains in the pellet. All other polysaccharides are hydrolyzed. Cool samples in the block on ice to room temperature or cooler, then centrifuge the samples at 10,000 RPM for 15 minutes. After centrifugation, discard the supernatant, ensuring that the pellet is not disturbed and no material from the pellet is removed. For this purpose, leave approximately 150 microliters of the supernatant in the tube. Wash the pellet several times with water. Next, perform a Sayman hydrolysis with the concentrated sulfuric acid in order to completely hydrolyze the crystalline cellulose pellet into glucose. Start by adding 175 microliters of 72% sulfuric acid to the Sardsted tube and incubate for 30 minutes at room temperature. After the initial incubation, vortex the sample and incubate for another 15 minutes. Then, add 825 microliters of water and vortex, and centrifuge the samples at 10,000 RPM for 5 minutes. There might be some brown insoluble material, mostly lignin, remaining in the tube, while the glucose is in the supernatant. The glucose content of the supernatant is assayed using the colorimetric anthrone assay. This assay is performed in a 96-well polystyrene microtiter plate. Prepare a glucose standard curve in duplicate, following the instructions in the accompanying written protocol. Prepare the samples by combining 10 microliters of each sample supernatant and 90 microliters of water into separate wells of the same microtiter plate as the standard. Finish by adding 200 microliters of freshly prepared anthrone reagent. Next, heat the plate for 30 minutes at 80 degrees Celsius in an oven with an aluminum heat spreader. Observe that the glucose-containing samples turn from yellow to blue-green. When heating is complete, let the plate cool to room temperature and shake thoroughly. Read the absorption at 625 nanometers using a microtiter plate reader. The content of glucose, the building block of cellulose, is calculated based on the absorbance compared to the standard curve established on the same plate. Here are some representative results of the polysaccharide analysis of a piece of poplar wood. The gas chromatogram peaks are identified by mass profiles and/or retention times of standards. The monosaccharides are quantified based on standard curves. The sugar analysis of the matrix polysaccharides shows a high abundance of xylose, representing hemicellulose xylan, but other sugars are also present. According to the analysis, 40% of the poplar material consists of crystalline cellulose. Please note that these contents and compositions can vary from plant to plant or between different plant anatomies.
We have just shown you how to determine the polysaccharide content and compositional analysis of lignocellulosic biomass. For the analysis of polyphenol lignin, tune in to part 1. Thanks for watching and good luck with your experiments.